Hi, I'm Greg Herbert, and I'm a paleobiology and malacology professor at the University of South Florida. The Sanibel-Captiva Shell Club has been a strong supporter of student research at USF. That support has allowed us to do more research on mollusks, but it's also given our students valuable experience. Thank you. The research we do at USF is about the conservation of marine mollusks, especially species of Florida's Gulf Coast. There are many lists of species that live in this area, but some probably haven't been seen alive by humans in more than a century. Some are probably only known from dead shells. So one of the projects my lab is working on is to figure out which kinds of mollusks are out there now and how they're doing. This kind of work is difficult for many reasons. It's hard just to know what's on the seafloor 1,000 feet down and 150 miles offshore anywhere, and we're attempting to do that everywhere in the eastern Gulf of Mexico all at once. So how do we do that? For the last 12 years, we've been fortunate enough to run annual expeditions using research vessels from the Florida Institute of Oceanography. Those vessels can go far offshore for many days, and we have equipment that can grab large samples from the deepest parts of the West Florida shelf and bring it up to the surface. I take up to 10 students on each trip, and we live on board the research vessel for 4 days each trip while we do our work. We usually see amazing sea life like dolphins, porpoise, sharks, and sea turtles that come near the surface. Some of the deep water shells that come up, Cyratus boei. We have a slit shell called Paratrochus amabilis. We have a relative of the famous Junonia. This one's called Scaphella dubia. This is a distant cousin of the tulip snail. This one's called Halprinia temessa. This little murex was named after the conservationist Rachel Carson. This is a beautiful cone called Conus mazai, and a very strange relative of cone snails called Polystyra albida. Those are just some of the examples of the diversity of life that lives in the deeper parts of the Gulf of Mexico. One way we try to reduce the impact that our sampling has on the environment is to take mostly dead shells. We know that shells that still have their original color and luster are relatively young, because those features are the first to go when dead shells are left out in nature. We use the dead shells instead of taking more live animals. Here in the lab, we clean shells to get rid of the mud and sand, and that's followed by months of work sorting and identifying each one. We don't waste any shells. We use even broken pieces of shell down to 5 millimeters because that one 5 millimeter piece might be the only record we have for some of the rarest species. Once we're done, we use statistics to predict how many species are actually out there at each site we sampled, and then we visualize those predictions using heat maps, where hotter colors indicate more species. Our goal is to show where the most productive and diverse shell-producing areas are out in the Gulf, so those areas can be considered for conservation. Proper management of shell producers is so important because mollusks actively build reefs and hard bottom that provide food and refuge for thousands of other species. We have another half year of work to do on that map as we check and recheck all of the identifications and statistics that go into it, and we'll have more updates for you all soon. Thanks again.
Hi everyone, I'm Jamie Rogers, a PhD student at the University of South Florida studying applied anthropology. I'm an archaeologist who uses shells and animal bones from prehistoric sites to investigate the human-environmental relationship over time. And I do this to better understand the impact that humans place on natural resources today and in the past. I hope to pursue a career as a researcher at a university or with the National Park Service after graduation in a few short years. And I want to thank the Sanibel-Captiva Shell Club for funding some of my research, which compares the growth of historic and modern oysters from the Tampa Bay estuary. This is important to investigate because growth and size data are used often in oyster management. And the growth and size of modern oysters has already been changed by overharvesting, pollution, and warming temperatures. And scientific collection of these data only goes back to the 1950s. Possibly missing important times of declining health of the species. I use information on oysters from historical records, such as newspapers, archaeological shell middens and mounds from before European arrival, and even fossils, because they offer a better baseline for oyster health. A recent study from my lab, comparing modern and archaeological oysters from Crystal River, showed that oysters today are much smaller in size than they were 1,000 years ago. And the loss of these colossal oysters affects how well modern oysters filter seawater and protect coastlines from storm surge compared to before. My archaeological fieldwork in Tampa Bay has recovered oysters measuring over 7 inches long about 1,000 years ago, and thus far seem to echo the finds at Crystal River. The Sanibel-Captiva Shell Club funded part of my research that studies growth rates of oysters. Shells grow like trees, in incremental bands, and the chemistry of each new layer of growth changes with those in water temperature and salinity. In the lab, we can measure these chemical differences, interpret the seasons, track the growth and age of oysters, and we can do this by counting the seasonal water temperature changes that are preserved in the shell. And we do this at a very small scale, like less than a millimeter. And we can incorporate other chemical analyses to look at different elements in relation to storm events and pollution. And just to give you an idea, here I have an oyster from about 1,000 years ago in Tampa Bay from an archaeological site. And you can see how the height of it and also the depth and the size of the hinge. Here I have an oyster from Fort Brooke from the 1800s. So you can see some changes in size, and also the hinge is pretty different. And then we have one from 2020. So very different in terms of size. And these all have cascading effects in the ecosystem. So what we do when we collect oysters is take them back to the lab. We look at the hinge, this part right here. And what we do is cut this in half down the center. And that exposes the bands. They're alternating white and gray. And that's related to summer and winter growth. So we can count those growths to better understand age and measure them to understand growth rate. And then we can incorporate some isotopes and elements to better understand chemistry of the water. So the historic oysters I just showed you were recovered from the Fort Brooke archaeological site, a military outpost from the 1800s located in modern-day downtown Tampa. And this study will help quantify how much oyster growth and size has changed over the last 200 years and extend the current scientific baseline used by scientists in Tampa Bay by over 150 years. So far, I have measured about 1,000 oysters from Fort Brooke and several hundred from earlier archaeological contexts. I've also submitted permits to retrieve the modern comparative samples, and I'm making headway in getting the samples ready for stable isotope analysis and plan to have that done in the near future. The goal is to submit the final work for publication in a peer-reviewed science journal and share the results with environmental land managers and organizations doing oyster restoration in Tampa Bay today. I'm eager to share my results with the Sanibel-Captiva Shell Club and want to thank them again for their contribution.
Hi, my name is Tomlin, and I am an undergraduate student at the University of South Florida, finishing my bachelor's degree in environmental science and policy. Also, I am aiming to go to graduate school for horticultural sciences.
Hi, I'm Nicole Seiden, and I'm a graduate student at the University of South Florida. I'm finishing my master's degree in environmental science and policy, and I'm aiming for a career in oceanography.
We would like to recognize the Sanibel-Captiva Shell Club for funding our research on estimating the lifespan of Florida's horse conch, Triplophysis giganteus, and determining their need for conservation.
These animals grow by adding new layers to their shell lip. And as these new layers form, the chemistry of the layers will change in response to changes in temperature. Using the funds from the Sanibel-Captiva Shell Club, we were able to sample these layers to recreate the history of temperatures that these animals experienced, and from that, the number of years that they were growing.
We used 2 of the nearest record-sized shells for our analysis, and our results demonstrate that Florida's horse conch is longer lived and slower growing, with a lifespan of about 14 years.
So let me show you some results. Here is what we call a profile. Now this x-axis represents the shell length, or how large that shell is. And each of these peaks is going to represent a summer temperature, whereas each of these valleys, the lower points, are going to represent a winter. Now, as we said, these shells grow by adding new layers right to their shell lip. And as these layers are growing or forming, the chemistry is dependent on the ambient water temperatures. By plotting the temperatures here, we can now count the number of summers or the number of winters. But you see how this lip gets very dark? What's going on here is the growth layers are very narrow, and our method of sampling is not detailed enough to get in between those narrow layers. So we had to adopt another method, and that's called laser scanning. And that was just so that we could get a little bit closer and more detailed results. Growth in animals generally follows an S-shaped pattern, where the growth is slow at the very beginning and the very end of life, and generally rapid in between. Horse conchs do this too, but they're unusual in that they grow rapid the majority of their life. It's how they get to be so big. Now that we have the age of these shells and a growth pattern model, we're able to estimate that the age of the maximum size shell at the Bailey Matthews National Shell Museum, which is 60 centimeters long, lived for 20 years. Now, we still have a little more work to do, but we've estimated the maximum lifespan of the horse conch to be about 25 years. Once we finalize our results, we're planning to publish them in a peer-reviewed science journal. Thank you so very much for your time and for your support.
Thank you for your time and supporting our research.